Ever wondered how scientists can zero in on a single gene out of billions of DNA letters? Let me introduce you to one of the coolest tools in genetic engineering, zinc finger proteins or ZNFs. These are not just another lab gadget. ZNFs are like the Swiss Army knives of gene editing, and they're changing the way we modify DNA, one base pair at a time. Here's how they work. Imagine a tiny protein hand, where each finger can feel for a specific three-letter sequence in your DNA. Now link a bunch of these fingers together, and you've got a super precise tool that can hunt down nearly any gene you want. But ZNFs don't just find the spot. They bring along a molecular scissor called foci. When two ZNF complexes lock onto nearby sites, their foci halves meet and snip the DNA exactly where you need it cut. What happens next? The cell senses the break and rushes to fix it. That's where scientists come in. We can let the cell patch things up in a way that breaks a gene, useful if you want to turn something off. Or, we can supply a new DNA template, so the cell swaps in new genetic information with surgical precision. So why use ZNFs? First, they're super specific, meaning fewer off-target surprises, which is a big deal in gene editing. They're also compact, making them easier to deliver into cells compared to some other tools. ZNFs have already been used in everything from boosting crop resilience to engineering immune cells that fight diseases like HIV. But let's be real, ZNFs aren't perfect. Designing them takes time, skill, and honestly, a decent budget. Not every DNA sequence is easily targetable, and sometimes, getting high efficiency is like trying to win the lottery. Success rates can vary a lot. Still, the tech keeps getting better. Thanks to AI and smarter design platforms, creating custom ZNFs is way more streamlined than it was even a few years ago. And with new delivery methods, scientists are pushing the boundaries of what's possible, both in the lab and in the field. Even though CRISPR grabs a lot of headlines, ZNFs still have a solid spot where you need pinpoint accuracy and a small footprint. So next time you think about gene editing, remember the mighty zinc finger proteins. They're proof that when you blend nature's blueprint with human creativity, you get something truly remarkable. Thank you for watching.